All right, welcome back, everyone. Nobody realized this, but you're all part of a larger experiment now. We're going to warm up the temperature in the room, and those of you that didn't get lunch versus those that did, we're going to see how you respond.
I think it's just the speakers that are going to pass out.
All right. I just wanted to, again, say thank you to the backdrop of YIBS who helped to organize this event. They've done an incredible amount of planning and provided support for this event. So a big thank you to those. They're probably not in the room, but make sure if you see someone on your way out, thank them for all of their hard work on putting this event together for us. I also want to note that I failed to mention at the beginning of the day, there is a slight change to the program as it was originally announced. So we're going to have a different speaker for the final slot. On the original slate of speakers, we had Shane Campbell-Staton, who unfortunately isn't available to make it today. But we called in a fantastic pinch hitter for the afternoon, and I'll elaborate more on that when we get there. So just put that on your radar. It's my pleasure now to introduce Eric Riddell, who's visiting us from the University of North Carolina at Chapel Hill. Eric's a physiologist. I'm going to call you a physiologist.
That works.
Is that fair? Who examines the function of organisms in the thermal environment. His research blends experiments, models, simulation models to identify how physiology shapes individual behavior, through all the way to species distributions, adaptations, and extinction risks. He's done some fantastic work, including things even like mammals and birds, which we don't have a lot of coverage for here in our insect lizard more sort of focused group. But we're excited to hear about your.
We also haven't heard about salamanders.
Salamanders. So, okay, fair enough. Thank you.
Yeah.
All right. Thank you.
Thank you so much. I'm really excited to be here today. We'll talk a little bit about prediction and ecology. We're going to talk about landscapes as well and what role landscapes play in our understanding of responses to climate change. And pretty much, I think everything in this talk is not published. So these are kind of the things that are on the forefront of my mind, things that are coming out of our lab, hopefully soon. This is a video of the Mojave Desert. This is at the bottom of a sort of ancient lake that has since dried up. And really what I'm trying to evoke here are images of hot dryness, but also of topography. We see here there's not much on this lake basin. It's very flat. There aren't many places to hide. However, behind us, there are these magnificent mountain ranges with lots of topographic relief and shade. And that's one of the attributes of the environment that I want to talk about today. So I study ecophysiology, which I don't really differentiate between physiological ecology, but I define very broadly as the study of how organisms function in their own environment. And one of the questions that my research program really focuses on is this one, which is how does the niche shape species responses to climate change? And specifically, how can we model the niche from a physiological mechanistic perspective? As people have talked about today, there are a few ways that this is done. Probably the most common way that it's done is using correlative niche models, climatic or otherwise known as climatic envelope models, where we take presence locations and sometimes presence and absence locations, and we combine these with different layers of temperature and precipitation, different axes of environmental variation, to generate some sort of statistical model that helps us predict suitable habitat for this species here that's widely distributed. It's for a tree swallow. And how those combinations of climatic conditions will change under future warming scenarios. As someone who is, sort of deeply rooted in organismal biology, there were a lot of issues that I sort of ran into with correlative niche models, which are here below, which I'll touch on very briefly, species interactions, right? So a lot of these presence locations reflect not only the abiotic environment, but also the biotic environment, and it's extremely challenging to tease those apart. There's also the problem of non-analogous climates, which is that climate change is going to produce combinations of climatic conditions that don't exist today. And so statistical associations won't be able to tell us if a species can persist there or not based upon correlations. And there's also space for time substitution. So we're assuming that the relationships that we're developing across space are also going to hold for what we might see with time as well. And there's a lot of literature coming out in the past few years showing that this actually kind of breaks down and may or may not actually be true, that the relationships with temperature, for example, across space will be the same for what we see across time. Now, this is sort of this fundamental problem that we have. In correlative niche models, we have these two pieces of the puzzle, right? We have the organism and the environment, and they use statistics to link those two things together. My research uses biophysics as sort of the fundamental mechanism that links an organism to their environment, and we do so by largely downscaling macroclimatic conditions to the microclimates that organisms live in. This takes a really deep understanding of all of the biophysical processes that are involved in heat transfer so that we can ultimately predict a steady state body temperature of our focal organism of interest. This is a woodland salamander. This is one of the species that I study, the gray-cheeked salamander. They are this cute, yes. And they live in the southern Appalachians in the mountains. And they've been a really great system for me to help understand the physiological mechanisms that are shaping their geographic distribution. Now, the reason we're interested in body temperature, as everyone has sort of been echoing this whole morning, is that body temperature is related to performance. So we can use the thermal performance curve and reproject it back onto space to make predictions about where we should actually see this particular species in terms of its distribution and how it might change with changing climatic conditions. So for me, biophysics is this missing piece. Instead of statistics, it's using math and heat balance in order to understand organismal performance. I don't just study salamanders. You can do this with all sorts of organisms. So I've been working a lot with birds, small mammals, lizards, amphibians, insects as well, where you can use these first principles to actually understand how much heat an organism is either gaining or losing to the environment, and then whether or not an organism actually turns that into a change in body temperature or resists that change either by metabolic heat production or evaporative water loss if you're an endotherm. So you can use this exact same set of physical principles to predict how organisms respond to environmental change. The other component that I like about this is that all of us probably in some form or another got into this business because we are interested in organisms. We are interested in the traits that they have, the phenotypic variation that there is in this world. And this framework allows you to explore that. So these are just highlighting some examples of some of the traits that we use. For instance, how much solar radiation the feathers absorb, or what the thermal preferences are for certain lizards. These help us to understand how hot animals get, how much energy they're burning, and how much time they have for activity. So these are just some examples of how we can use these models. Now I want to talk about the landscape now. So we've talked about kind of the general philosophy, but what about the environment? Again, the environment is really complex. And I think one of the things that we focus on, Jen just had a question about this, where do we stop with complexity? I think that's an important question that we need to ask ourselves. When and where does complexity matter? And in this case, I want to focus a little bit today on topographic complexity. I would say that this philosophy allows for really precise predictions. Whether or not they're accurate enough is up to us to explore. So one of the first things that we had to do was we wanted to understand how shade develops across a landscape throughout the day. So I worked with this really talented undergraduate, David Adams, to develop this algorithm that essentially measures the angle of the sun relative to the topography of the landscape anywhere in the world. This was for two purposes. One, to estimate cast shadows, so how a mountain might cast a shadow on another mountain behind it. And a second is for shaded relief. So this would be self shadows, so shadows that are cast by yourself, like this one right here, or that one. It's essentially determining the zenith angle, which is the angle directly overhead, relative to the altitudinal angle, the solar altitude. And basically by comparing those values, you can determine whether or not an area is in shade or full sun. And that has obvious implications for how hot, let's say, a lizard or a salamander might actually get on the surface of the landscape. So what you're looking at now is a map of North America mostly. And what you're seeing here is the annual solar radiation in this color scheme, where really bright colors are higher amounts of solar radiation. So what you'll see is that at lower latitudes and at higher elevations that we have higher amounts of solar radiation. So that's just basically what you can see. And all we wanted to do was compare two simulations where we said the whole North America is flat versus let's incorporate the amount of topographic complexity and shaded relief that we have. So now what you're looking at is the change in solar radiation across all of North America, where these brighter colors are meant that we underestimated how hot they would get, and the cooler colors mean we underestimated how hot the landscape would get. So you can think about it this way. These are actually, the brighter colors are hotter than expected, and the cooler colors are cooler than expected. If we zoom in to the Rocky Mountains, we can actually see what's going on here a little easier. So the southern slopes are getting hotter than we expected, and the northern slopes are actually cooler than we expected if it were a completely flat landscape. This makes sense. We know that the southern side of your house, for instance, gets more sun and is generally warmer than the northern side of your house. And so this is exactly what we would expect. But of course, the topography is not the only variable in this simulation. We also need to think about forest cover as well. So this is a map from the US Geological Survey showing the amount of forest cover across the lower 48 states in the US. So I'm just going to overlay this on top. And I want to sort of give you a couple takeaway messages here from this part of the talk, which is if we look at the eastern part of the US, and we're looking at the Appalachian Mountains here, we can see this effect of over and underestimating how much solar radiation the sun gets. If we add forest cover on top of it, it's almost completely masked. So the amount of forest cover that we have almost makes this topography component not matter as much. So this part of the complexity may not matter. We'll demonstrate that with some simulations with actual organisms. On the West Coast, this is without forest cover. If we add forest cover here, we actually see that a lot of this over and underestimation of topographic complexity persists. That particularly at higher elevations, you tend to see less of this forest cover, and a lot of the effects of topographic relief persist, and suggest to me that this would be an important part of the world, these highly mountainous with less vegetation, or less vegetation in general, let's just say if it's hot and dry, to incorporate or to think about topographic relief and shade. So let's actually do that. So what I'd like to do now is pick some study systems where we can explore this idea. So the first study system that we're going to talk about is in the Mojave Desert. And the second that we'll talk about is in the Southern Appalachian Mountains. These are kind of like polar opposite environments, like a lot of water and trees, basically no water and trees, hot, cool. And they also have very different organisms that live in these environments and that are sort of more abundant in these environments. If we think about the deserts of, or the arid regions of the American Southwest, we may think often of lizards. This is a Scaloporus occidentalis, the western fence lizard. And so we're going to be modeling how activity and energetics might change if we include or exclude topographic relief. And on the East Coast, we're going to be thinking about salamanders as well, because this is the global hotspot of salamander diversity. There's an extremely high abundance of species in this area. We're sort of interested in how does topographic complexity affect our predictions of changes in activity and climate vulnerability for these species. So we're just going to jump right into it. What we did was we conducted these simulations incorporating topographic complexity or ignoring it, and we compared them. What I'm showing you here on the left side is just a three-dimensional landscape of Daytime operative temperature, which is the steady state body temperature for this lizard. I have Death Valley here that's indicated here by this DEVA. So that's Death Valley. You can see it's quite hot. If we look at the change in body temperature, the first thing I want to point out is that you can see topography. If there were no differences between the simulations, it would just look like a blank square. There would be no, you wouldn't be able to see anything. So the fact that we see any topography means that there are differences in body temperature across the landscape, anywhere from 12% to minus 36% difference in body temperature. What about for how much energy they're burning and for how active they are? We see that if we exclude topography, that we either under or overestimate metabolic rate by 13 to 21%. And similarly with activity, we under or overestimate it by 30% roughly. And you can see how much variation there is across the landscape. It's kind of like scattered about depending on the topographic complexity. So this would be a really fun map to go out to these sites and test whether or not, activity is higher or lower than you would expect. So for lizards, it seems to matter quite a bit. This is just a graph that sort of summarizes these effects that we see these effects predominantly on northern and southern slopes. So if we assume it's flat, we are going to over-predict how much energy they're burning on northern slopes, and we're going to under-predict how much they're consuming on southern slopes. We also ran some climate change simulations for these lizards as well. They obviously burn more fuel under future scenarios. They get hotter. One thing I like about this result here on the x-axis is slope of the terrain, and on the y-axis is metabolic expenditure. And you can see that under future warming scenarios, they're burning more fuel at the same slope. So, and that's because that they're actually further up on the steeper part of the thermal performance curve of metabolism. So what I think is kind of cool about this, it kind of reminds me about Jensen's why mountain passes are higher in the tropics. Are mountains steeper under climate change? It kind of has this like really sort of interesting idea there. So let's look at salamanders, which are very different. So now we have steady state body temperature, which includes the effect of evaporation here across the landscape. And we see that we have a little bit of deviation in body temperature estimates between 13 and 12% by ignoring or including topography. And but there's really almost no effect of the change in metabolism or activity. We overestimate by 2%, underestimate by 5%, and then basically no change for activity. So for the salamanders, and so why is that? It's likely because they live in these forested communities where a lot of solar radiation isn't reaching the soil surface where they are. They're also nocturnal. So the point of this is to really sort of point out areas where, and of the tree of life, as well as across the globe, where these types of considerations are going to be really important. And this is actually just the same result as we found for lizards, where we over and underestimate on northern and southern slopes. And, but the magnitude is much smaller. So this is kind of one of the takeaway messages for this analysis. On the x-axis, we have aspect, so the way that the mountain is facing, basically. And on the y-axis, we have either the change in metabolism or activity. If you'll note for salamanders, which are in this sort of lighter color, they hover around 0. So it really doesn't matter. If you exclude topography, you don't get really big differences. For lizards, there's a lot of variation. But the cool part about this is that if you average it, it's basically 0. So if you were to run this analysis and say, well, does topography matter? Well, on average, it doesn't matter. But if you look at the variation, you could be off by anywhere, especially for activity, by 25 to 30%, either over or underestimating. And if what we're interested in is the variation across the landscape and making these more fine-scale predictions, then I would argue that this is really important. Even though the average is 0, it's the variation that we're really interested in. So I'm really excited to continue to extend some of these models with Martha Munoz. We have a really excellent team of Martha and Nathalie Alomar and Izzy Berger, who's my student. Nathalie is in Martha's lab. Nathalie has been working extremely hard to collect physiological data from this group of really diverse salamanders. And Nathalie, I'm sorry, I don't have a video of you doing this, so I inserted a video of myself doing this. But this is basically what Nathalie was doing, where we would she would go out and collect these salamanders all across the country and bring them back into the lab and measure their metabolism and their water loss. And they did this across how many species now? 45, 40, 30, 30 something? It's a lot of species. I'll show you that in a second. And essentially what we're trying to do is understand how physiological diversity in this particular group of organisms may be related to climate vulnerability. I'm going to check the time real quick so I have just 3 minutes. Is that right?
No, we started late.
Oh, we started late? Cool, great. All right, great. So what we do is and the reason this question is important, I want to say, is because these salamanders are kind of like this hallmark system of niche conservatism where they tend to all live in similar environments. They all live in these cool, high elevation environments. And so the assumption is that physiologically they are very similar. And I'm not going to steal Nathalie's thunder too much. I'm actually not going to show any of her data. But what we did is we took Nathalie's data, we incorporated it into these biophysical simulations that incorporate biophysics, physiology, so that we can predict activity and estimate how much food that they're bringing in, so we can estimate their fitness, how many offspring they can reproduce based on their energy budgets. And we do this across the entire landscape, so we can make predictions about where they should live. And then we verify whether or not these species live there based on presence data or geographic locations. And we do this all in our own custom-made mechanistic niche models in-house. So these are some of our preliminary results. I think Martha and Nathalie saw these like 2 days ago. So like these are like really fresh. But basically what the take-home message is, we have this phylogeny of the species that they've collected so far. And we have the change in energy consumption, which is being mapped across the phylogeny under climate change scenario, as well as the change in activity. And there's a lot of variation. Some species actually are predicted to increase the amount of activity they have. Others are more restricted. Some are burning more fuel, some are burning less. So based on the physiological data that she has collected, we would expect a lot of variation among species. Even though they are seemingly very similar, there are differences in physiology that could impact climate vulnerability. So the point of this, what I'm trying to tell you is that this mechanistic modeling that we're doing is really an excellent foundation for asking questions in ecology and evolution. It gives you a really strong foundation for prediction, and it gives you a really strong foundation for trying to ask these questions about niche conservatism, the phylogenetic distribution of vulnerability. So I'm going to take a break from that right now, and this will be the last part of my talk here, and this is about endotherms. While I was at Iowa State University from 2020 to 2023, we started a project on tree swallows. So this is the tree swallow up here. They are migratory, insectivorous passerine. They are a really charismatic bird and really well studied, which in some cases is hard because of like someone already did that 30, 40 years ago. But also you have tons of data to rely upon, which I'm going to show you here. So we studied these tree swallows for several years. One of the things that we did was we implanted temperature-sensitive pit tags, which you're actually seeing here, into their body cavity, so that we could, every time they visited their nest, we could understand we could get what their body temperature was, as well as how active they were, how many times they were provisioning their young. These are images from some of the camera traps that we placed up there. So we had 2,222 readings of activity and body temperatures on 46 tree swallows over the past 3 years. And what we were really interested in here, what kind of brought this question was, how can we study thermal performance curves in endotherms? It's actually really challenging. And Katie and Danielle Levesque have posed this question, I think in 2021, too, something like that, in JEB. And it was a great article. And it got us thinking about, well, how can we actually sort of model thermal performance curves in endotherms? And what's like the relevant x-axis here? Because it's not body temperature. So what we did was we took weather station data that were out at where these tree swallows are kept We combined it with some morphological measurements of how much solar radiation, that's obviously not a tree swallow, and we incorporated into these biophysical simulations that I call Endoscape, and to estimate how much metabolic heat these birds need to generate and how much evaporative water loss they need to lose to maintain a stable body temperature, which is what you're seeing here on the far right. So time of day on the X, and something called net sensible heat flux on the y-axis. So this is how much heat they either need to gain or lose to maintain a stable body temperature, okay? Now, our idea was to actually plot performance against net sensible heat flux, because it's not body temperature that's changing, and air temperature certainly isn't a really good surrogate, but net sensible heat flux is something that endotherms respond very readily to. So they need to increase metabolic heat production and evaporative heat loss in order to stay in to maintain a stable body temperature. So what you're looking at here is really cool. On the x-axis is net sensible heat flux, and on the y is relative provisioning rate. Now, they don't provision a lot at cool temperatures because they're provisioning in June, so cool temperatures don't really exist. But we see this peak at 2 watts. Now, here's the part that's really cool. We could bring in data from estimates of how much heat they're burning and how much water they're losing during provisioning. And when you add the metabolic heat production and evaporative water loss to this peak, you see that this peak in performance, if you add how much heat they're generating, they generate tons of heat while catching insects and provisioning their young, you get 0. That's a stable body temperature. So their peak for provisioning occurs at a heat loss that is, to us, quite cold. This is actually about 14 degrees Celsius. That's their peak temperature for provisioning their young. But they do so because they're generating so much heat that in order to keep a stable body temperature, that's where the optimum is. Now, our goal was to actually predict, can we predict the breeding biology of this species? So what I'm going to show you is this GIF where we mapped those optimal conditions across the whole landscape. So optimal for provisioning is in yellow, suboptimal is in blue. And we're going to play it for every hour of an average day of that month. So this is the sun rising in April, sun rising in May, sun rising and setting in June, sun rising and setting in July. And so we were able to model and track the optimal conditions for where these birds should be able to provision their young. And so here's the cool part. This is in June when pretty much all of the provisioning happens. This is eBird data showing you where this species persists across North America. And this is our model's estimate of where they should be based on what's optimal for provisioning their young. And the little dots are where eggs hatch. So that's data we have for where eggs have actually hatched. So the model works pretty well. And this is June under a warming scenario. So there's around a 15 to 20 degree increase in latitude in terms of where they should be. These are tree swallows. You can talk to Jen and Katie. There aren't many trees up there. So that's a pretty big problem for tree swallows. And this is just showing you sort of like we modeled all April, May, June, and July. And you can see that our model does, so this top row here, so much data. I was like, do I show this? But yes. So this top row is showing you when tree swallows lay eggs, mainly in May, and then when those eggs hatch and fledge, mainly in June. This next row here, and each column is April, May, June, July. This next row here is showing you eBird data for where those birds exist latitudinally. This is all regressed against latitude. And these are our predictions for current, and these are future. And so you can see that our model does really well at predicting peak times when these birds should be provisioning their young. And of course, we get really big shifts in optimal conditions for provisioning young under future warming scenarios. And these are just the maps showing the same thing. So what I want you to take home from this talk is that mechanistic niche models, they're really complex. And I like complexity, but I also understand that it comes at a severe cost. But I do think that these models can tell you when complexity does and doesn't matter. And that's important. It depends greatly on the context of the environment and the organism. And then I'll just reiterate that I think this provides a really solid framework for understanding broad ecological and evolutionary questions. So with that, I'll just like to thank the following people and funders, collaborators, and take any questions if I have time. Yes, Jen.
That was really exciting, cool. I think my question is, do you just connect? Have you tried connecting the two, or is it too fresh? Like, what would the tree swallow prediction in the future look like in the shaded parts of the mountains?
I've thought about the topography component. That is less of an issue because they're not ground dwelling. And for the most part, they tend to, that's a good question. In general, where tree swallows are concentrated doesn't have much topographic complexity. That's not always the case. There's some places in California. So my guess is that it wouldn't change things tremendously based on their current distribution where they're located. Yes.
One of the things I was sort of thinking about in the context of some of the questions that I posed, which is, around plasticity and how important is plasticity.
Right.
One of the things we've been thinking about a little bit is, can we use things like mechanistic niche models to explore, like, how important is plasticity? Does it make a difference, right?
Totally.
And I think this is more of a comment than a question, so I'm very sorry, but I think the other thing that mechanistic niche models can do is help us exclude some things, or, say, actually, this is really important.
Totally. Yeah, I've done quite a bit of work on physiological plasticity with metabolic rate and water loss rates, and showing that, like, based on empirically what we see they can do, it matters a lot. And for the salamanders, it was the difference between simulations where they're completely extinct to where actually energy balance increased in the core of their range. Yeah, it matters a ton. It's a great tool.
Oh, thank you.
Yep. Yes, Ray.
I was really interested in phylogenetic patterns that you were saying, and thinking conservation managers able to take advantage of that to see which populations might be worth salvaging and which are just lost cause.
Yeah, that's it's basically what I've learned right now from speaking with conservation managers, is that we speak different languages, and there really needs to be more of sort of a way for us to communicate. And in some ways, sometimes it's almost adversarial, because it's like, oh, you're out collecting animals and you're causing harm. And but I think that, my goal, especially being in North Carolina now, is to convince them, to show them, like, here's what these models can do, and here's what, we might be able to isolate, which populations you should be concerned about and which ones you shouldn't. The green salamander is actually a great example, because it basically has a tiny population in North Carolina that is at high risk of going extinct, basically because they are specialists in terms of the habitats that they live in. They really like granite, and they really like trees. And so I think that some of these simulations could help in things like assisted migrations, potentially, as well as, certainly it's not a fun conversation to have, like, you shouldn't care about this species because, like, my model says it's gonna, that's a lot of pressure on me. I think that we should more use them as tools to understand process. And I think maybe that's where it gets lost, is because they're like, I want a map that's going to tell me where they're going to die and where they're going to live. And it's like, well, I can tell you how these processes might influence that, and I can give you my best guess, but it is hard, when someone really holds you to it and says, well, I'm going to go buy, $3 million worth of land because of this. Is that, are you willing to put, money down there? It's a hard question. Yes.
Can you flip this around and say, for the species to survive in its present range or part of its present range, its body size or its metabolic rates will have to evolve?
Oh, yeah.
A certain amount?
Totally. So that would be, you could basically take the phenotypic space that we have, see if there's any in that existing variation that is capable of maintaining, and say, like, yeah, birds should shrink by 30%. And if they do that, this part of the population persists, they can stay here. So you could totally do that. The problem is things like body size or feather coloration. Like, I've actually run this analysis. How light would birds have to get to reflect the amount of solar radiation so that they maintain the same heat flux under future warming scenarios? And it's around 30%. That's a big, big change. It's actually phenotypically possible based on the amount of variation for some species. But you have like issues with like camouflage. Like, so now I'm really light and I'm reflecting all the sunlight, but now I stick out. And so that's a big problem. Or now my mate doesn't want to breed with me because I'm a different color. So it's complicated. It's body mass, you could argue the same thing. So like you could totally explore those questions with these models. But it's hard to know what all the trade-offs are. I didn't see, yeah.
Can these models also predict, for example, a possible isolation of two phenotypical groups, where you can have like lighter ones just like taking south, bigger ones now fully, like at some point if you want like this for enough, you see this population kind of, like, is this possible to test?
So I do think Lauren Buckley has done some, and Joel Kingsolver have done some really excellent work where they integrated biophysical niche models with genetic algorithms, and in order to understand like, can species evolve given certain heritabilities that we're estimating, like is phenotypic evolution possible? To what extent can evolution like rescue this population? That's kind of what you're asking a little bit. Like, absolutely, yes, if you have populations with different phenotypes, you could certainly simulate where they might live. But then you'd have to incorporate some sort of genetic algorithm but since these are based on performance and fitness, like it's a really great synthetic framework for operating across from organisms to ecology and evolution. So absolutely, yeah. It's my career grant that I'm trying to get funded. Yes.
Yeah, thank you so much. One thing I was wondering is the Western United States is another mountainous sort of above and showing this sort of beautiful, I guess, more pronounced pattern, right? I'm wondering if you have any thoughts on whether it's rock type, whether it's glacial still present, or things like that, would have small but
appreciable. Yeah, I mean that's a great. What is appreciable, right? It's if you study some rare moss that like lives on the granite that from receding glaciers, like this is like really important. But I think the downside to all of these models is that they require a lot of parameterization and they all require a lot of math. And so you have to be comfortable enough with, I liked math, but I was never thought I'd get into biophysics. But it ultimately ends up being such a cool tool that you get hooked on it and you're like, oh, it's like, you kind of see the matrix a little bit. So yeah. So the answer is yes. Like it can work on any scale that you want it to. I mean, my advisor, Mike Sears, he spent his career just thinking about lizards on a fairly small landscape and how thermal heterogeneity influenced lizard behavior on really small scales. And so I think absolutely, in any scale that of course, computationally it becomes hard.
All right.
Sounds good. Thank you.
All right, fantastic. So we're going to go from the land of terrestrial megafauna down to the land of aquatic microbes, right? It's my pleasure to introduce Mridul Thomas. Mridul has worked across the domain of theory and experimental ecology and I think has at this point published the largest synthesis of thermal performance curves of marine microbes that I know of to date. So he really has sort of worked across that entire domain. He's visiting us from the University of Geneva. So please, the floor is yours. Thank you.
Thanks very much, Dave. And thank you all for being here. So you're not going to see a lot of pretty pictures, so be happy now. But these are the sorts of organisms that I work with. What you're looking at is quite a diversity of phytoplankton. These have diverged over hundreds and hundreds of millions of years ago. So these are groups that are separated by deep evolutionary time. You have the cyanobacteria, you have green algae, you have diatoms, you have coccolithophores. So they're beautiful. They are very interesting to study. They also pose lots of problems for some of the questions we ask because, what's a species for some of these and so on. But I realized the first time I spoke in front of a temperature audience, I need to have a phytoplankton and not small lizards slide. but they kind of are. I mean, we mostly treat them like they are, but there's a few subtle differences. They don't have the ability to move, really, not much, and they can't escape the temperature they experience. The environments are homogeneous. So they're really at the mercy of the environment and the temperature that they are subjected to. On the one hand, it means that a simple weather station measurement gives you what you need, but it also means that small changes in the environment have huge biological consequences, especially because their rates change so quickly. And that's the other thing. We can measure population growth rates, which is really nice, because it means that when we're saying that something is beyond its maximum temperature, it means that their population is literally going extinct. And, well, they matter because ultimately they are responsible for about half of all carbon dioxide that's taken out of the atmosphere. So this NASA satellite image where you're seeing the phytoplankton in the oceans over here is showing changes of orders of magnitude on timescales of weeks to months. So they respond very quickly to changes in environmental conditions, and they have a huge effect on global biogeochemical cycles. So part of what we're trying to do is figure out how temperature affects them now so that we can understand what will happen to them in the future, so that we can get a handle on what's going to happen with primary productivity and carbon sequestration. It would be really convenient if this is all we needed to do that. Unfortunately, models, global ecosystem models that we rely on, do a pretty poor job of predicting primary productivity, carbon dynamics. This map is a little bit old, but even a more up-to-date publication has shown pretty substantial differences, like on the order of 20 to 30% in some of the global climate models, biogeochemical models. Here you can see, and there's spatial differences in where they differ. So here you can see the model makes a very different prediction. Well, this is an ensemble of models, makes a very different prediction in the tropical oceans than the observations do. So this is what we want to fix. And that's primary productivity, which is arguably the simplest thing that we could ask for from a model. We want to know more than that because these different groups I told you about differ in other important ways. The cyanobacteria are the only group of these that fix nitrogen. They're responsible for a huge amount of nitrogen being pumped into the biosphere. The diatoms are, for microbes, big and heavy, and therefore responsible for a disproportionate amount of carbon sequestration. Some of the other groups are good food. Some of the other groups eat other groups. So we want to know more than primary productivity, and primary productivity itself we do a bad job of. And we do have these models where you can see different groups of phytoplankton, and we make predictions about where and when in the oceans we see them growing. This is a group at MIT. And we see succession of different groups of phytoplankton. So there are clearly environmental patterns here, but there's important pieces missing in this environmental dependence. The one I'm most concerned about is the fact that interactions between abiotic variables are largely ignored in these models. So let's step back for a second. We understand how population growth rates change in response to single environmental dimensions. You've been hearing about thermal performance curves today, a lot about them. But we have analogous versions for nutrients and for light. And there is a similar level of generality to these. All photosynthetic organisms, so plants as well, should follow this curve. Essentially, every organism obeys this, whether they're a bacterium or a blue whale. So the generality is very conducive to mathematical modeling, very conducive to linking experiments with theory. And the parameters that we can use to describe these functions tell us a lot. You've heard about T-opt, you will be hearing more. There's also these half saturation constants or affinities, optimal irradiance levels, and more. And those parameters can tell us a great deal. If we focus just on the temperature performance curves for the moment, if you look at the optimum temperatures of about a couple of hundred phytoplankton strains, species, thereabouts, you can see that these optimum temperatures change pretty consistently with mean annual temperatures. There's some variation there, and there's some saturating here, which potentially needs some explanation. But overall, the general pattern from the poles to the tropics is quite consistent with what you'd expect from a theoretical model. This and quite a bit of other work was done with Colin over here. I had a photo of him, but then it turned out he's showing up today. So I had to quickly check all the work I was doing, and I will just point. But they can not only tell us about their evolutionary history, these curves can tell us where species are found today. So Maike Vogt, a scientist at ETH in Zurich, in Switzerland, has compiled about 20 million observations of species presence in the oceans. She's scooped up anytime anybody has gone on a cruise and measured the phytoplankton there. And now we can actually, for the first time, start to understand where phytoplankton are. So as I said, we can do experiments and measure population growth rates, but we have never known before this where the species actually are in nature. But now we have this data, we can start to fit simple machine learning models and ask those machine learning models, how does the probability of this species being present change as a function of temperature, which is what is shown here for two different species. And this is in some ways analogous to our thermal performance curve, but it's the species' temperature preference in nature. So the question that we are asking with Ting Liu, a visiting PhD student in our group, is how well can we predict this based on lab measured population growth rate based temperature performance curves? So if you estimate sort of a rough field optimum temperature and compare it with the lab optimum temperature, this is a one-to-one line, not a regression line, you can see it's not spectacular, but it's not bad at all, especially considering how noisy this data is and the fact that for the most part, we don't know what species are with phytoplankton. The fact that we have any correspondence at all is actually rather encouraging. So if you believe these curves tell us what species are adapted today and where they are found today, we can arguably start to ask questions about how diverse species ranges and diversity patterns will change in the future. So if we assume no evolution, which is unrealistic, but bear with me, then if we assume no evolution, then we find that a whole lot of species that can live in the tropics right now would die over there in the next 50 to 70 years, talking about 40 to 50% of them. Another way to read this is that there is evolution going on in the tropics right now, and we should be measuring this to understand what the consequences are. But the thing is, phytoplankton and species in general, they compete. And we think phytoplankton mostly compete for nutrients or sometimes for light. So this begs the question, how do temperature and nutrients work together? There's a long history in phytoplankton ecology of modeling competition between species for limiting resources. The resource competition theory was based on this. So we have R stars for these two species. We can make predictions based on dynamic models for when the two species will survive, which species will outcompete the other, or whether you will get coexistence. So there's a long history of this, but this has never been married to the temperature projections that we're worried about now. So we decided to try and do this. I'm not going to go into this model in much detail because frankly, I don't think you should take this as true mechanisms. For the most part, we have something like 100 different models of TPCs. It's different ways of describing curves, okay? So, but if you'll indulge me, you can think of this as two different, the effect of temperature on growth as being the difference between two exponentials. Let's call them birth and death. If you subtract one from the other, you can get the thermal performance curve. We already know that nutrient shape is over here, And let's say that nutrients don't affect deaths. That seems reasonable. So let's say that nutrients only acts with the ascending part, with births. And if you multiply it that way, and you make death just a function of temperature, you basically find one way of squishing together these two curves. And it turns out there's just not that many ways. What you get is something like this. So you get this landscape, this response surface of growth rate as a function of temperature and nutrients. And it's already been talked about a little bit earlier. Thank you, Ray. But the optimum temperature here is a saturating function of nutrient concentration. The Tmax and Tmin as well change with nutrient concentration and so on. And I think this is both interesting and rather important for everything we are trying to predict. So there's no real mechanism here. As I said, we are just drawing shapes. But we need simplified representations of physiology because we are not going to solve a physiologically explicit model in a global ecosystem model. So there is value to this simplification as long as it's consistent with data. And the point, I think, is to take from here is that mathematical constraints can take us quite far, even when we do not have much data. Also, Ray presented his metabolism-based model, which comes at this from a different angle, but mercifully makes similar predictions. So what are these predictions? Well, I told you that the optimum temperature is a saturating function of nutrients. This is data from 2 species. In our study, a later study by another group found the same for all the species that they measured. We're talking about differences of about 10 to 15 degrees in the optimum temperature at low nutrients over here. If you compile so the CTmax data from heterotrophs, the short-term experiments that you've heard a lot about from insects and fish and so on, and you will find there's broadly consistent patterns. If you starve organisms, the CTmax tends to decrease, though not always. But if you use longer-term experiments, as Brett did in the paper that Ray told me about 10 years ago when I showed him my data, you see much, much stronger patterns. So you see that optimum temperature decrease, you see that maximum temperature decrease. But though Brett's data is probably still the best that's available, which is perhaps a little shameful, we still see the same patterns in other organisms. So this is a heterotrophic flagellate, high food, middle food, low food. You can see, again, about a 10-degree difference in optimum temperature, decrease in the maximum as well. Mosquitoes, higher optimum temperature when there's more food, low optimum temperature when there's less food. In the interest of not believing my own ideas too strongly, I decided I ought to present the fact that this is not apparently universal. This is an iron-limited nitrogen-fixing phytoplankton, which perhaps increases its optimum temperature when starved a little bit. But essentially, we don't really know. It's still early days for this. We do not have much data, and we need more data quite urgently. So hopefully, I've persuaded you that this interaction exists. But assuming it exists, does it matter? Well, now that we can model population growth rate as a function of both temperature and nutrients, we can start to do interesting things. We can make dynamic models, exploring how a population will do in unusual changing environments. We can explore competition between species and more. So if you write just a simple chemostat model of population growth and just force it with a sinusoidal annual temperature cycle and ignore competition entirely, what you will see in the purple line is that warm seasons are much, much more harmful once this interaction is appropriately accounted for. So in the green line, which is the old model without this interaction, this species could do okay. The purple line, it will die for a large period of the season. That also means if you try and predict where this species will be found in nature, and you do it with the new model, you will find that it has a smaller species range. So the green areas are areas it's able to grow when you do not account for the interaction, but it cannot grow once you account for the interaction. So the species is more likely to die in warm seasons, and it has a smaller species range once you take this into account. That was just nutrients, but CO2 and light, as I showed you earlier, have a unimodal response. So there's an optimal light level and an optimal carbon dioxide level for some phytoplankton as well. And you would likely get something like this smooth triangular shape. And one interesting aspect of this is that whether you expect a species living in this hypothetical condition today, the black curve, whether you expect its population growth rate to increase, to decrease, or to stay roughly the same, is hugely contingent on the shape of the surface, the sensitivity, and uncertainty in both these parameters. So I think in order to make predictions for species, we need to grapple with this. There's more than that, though. Interactions will shape ecological interactions. So what you see here is a particular cyanobacterial species, Planktothrix. It's a toxic species, and a fungus that infects it and kills it in lakes, a chytrid. So a PhD student of ours, Joren, did experiments where he grew the phytoplankton species by itself with its chytrid host, with its chytrid parasite, and at a range of different temperatures and light levels. And he measured important rates of this interaction. I'm just showing the phytoplankton growth rate here. With that, we can model host parasite dynamics across temperature light surfaces. And you can see that there's conditions in which the uninfected host wins out completely. The chytrid cannot survive. But then once the temperature increases, the chytrid can start to increase and starts to experience huge and consistent fluctuations in some conditions. And you see different dynamical outcomes. You see regions where only the host survives, you see stable coexistence, you see limit cycles, and so on. So even in this simple system with just two interacting partners, the temperature and light interaction leads to this unusual landscape. So how useful is this? This is something we have to grapple with when we study phytoplankton, because there are species which matter a lot for the ecosystem, like some of the nitrogen fixers. But often what we want to know is the community response. So in ecosystem models, what is often put in as a temperature dependence is simply this exponential. So this is what is called the Epley curve. It is the maximum possible growth rate for the whole community as a function of temperature. And this, of course, arises as sort of the envelope of many temperature performance curves. So one thing that Colin has been doing is trying to extend this model of nutrient-based population growth rate to account for light as well, and also ultimately to try and link these population-based growth rates with community-based growth rates. So you have already seen this plot of the temperature nutrient growth rate surface. This is the same surface as a function of light, but only when the other one is ample. As you change both temperature and light, you get more unusual and jagged shapes in this present formulation. What does that mean? Well, to return to the optimum temperature, what this shows you is that the model predicts a saturating relationship for nutrients, again, but a unimodal relationship for light. And that is consistent with growth rates, optimum temperatures measured in the lab. But we can compare that Epley curve as well. We can start to think about how does light affect the scaling, the temperature scaling of growth rate. So this is a slightly complicated graph. What each panel represents is growth rates from phytoplankton cultures as a function of temperature within a particular band of light levels. So this is 0 to 10, 10 to 20, 20 to 50. These are micro-Einsteins per square meter per second. And this is, I've plotted growth rate on a log scale here. So the red line is that Epley curve, and you will see that its slope is changing. If you just look at that slope, what you see is that the temperature dependence of community growth increases quite steeply with light. I don't have much faith in this high point because of a lack of data, but it increases and either levels off or comes back down. We think it comes back down. Now, this is also consistent with that temperature by light by nutrient model. If you look at how the growth rate of the whole community, represented by the envelope over there, changes as a function of irradiance, changes as a function of, changes as a function of temperature. At high irradiance, it increases exponentially, but it is kind of flat at low irradiance. So this is what we see in field experiments of carbon uptake for the whole community. We see it from lab cultures of population growth rate, and it is consistent with what we get from a relatively simple model. So what are the implications of these interactions? Well, there's a few things I can think about. One is regions of combined extremes. So in the tropics, you have regions which are both the hottest parts of the ocean and the lowest nutrient parts of the ocean. These may be threatened by further increases, but they also may be valuable sources of genetic diversity because these species have adapted to present-day low nutrient and high temperature conditions. Of course, there's going to be composition shifts because of competition changes and migrations from high temperature to low temperature. But you may also see migration from low food to high food regions because that high food may allow them to survive the high temperatures. Related to that, species that do well at low food levels may be better able to tolerate warming because of that same interaction. And finally, the thing that I think we really need to grapple with is feedbacks, because by growing, they change the resource levels that they experience and therefore their own susceptibility to the environment. And there's no getting around the fact that we need dynamic models to sort of explore how this plays out. So we don't have a lot of data. Why is this not the case? Well, the usual answer in my field, at least, is that the experiments are too hard. You need 5 or 6 temperatures, you need 5 or 6 food levels, you need half a dozen replicates, and it becomes unmanageable. Now, I disagree with this, and so this is why this has got me interested in experimental design. This is work I've been doing with Ravi Ranjan over here. So in global change ecology, at least in the aquatic stuff that I work in, experiments are often done like this. So these are temperatures on the x-axis, nutrients on the y-axis, and the size of the points represents the number of replicates. So you have these present versus future designs. You have these ANOVA designs, which are still the most common designs used. And these experimental designs do not tell us much. We do not get much information out of that. Now, it turns out there's a whole world of experimental designs that are relatively uncommon in our field. The one that everyone knows and at least ought to try is a full factorial response surface design. You don't need the replication if you're going just like you don't need it for a temperature performance curve. But there's even more unusual designs. You do not need a fully balanced design like this. You can drop a bunch of points. You can use what is called an optimal design or a sequential or adaptive design coupled with optimal design. And you get these very, very strange shapes, which combine replicated and unreplicated points. Now, why is an optimal design so different from a full factorial response surface? That's because of the shapes we're talking about. This is that temperature nutrient response surface. All the points on the surface are not equally informative to a statistical fit. So this is on the right, a map of leverage. And you can see basically the points at high temperature and at low nutrients a little bit here contribute much more information to the fit. So you can drop a few points here, or you can let an algorithm choose where to drop them for you and limit your experimental effort, but still get a lot of information. So if you just run some simulations and compare a whole bunch of experimental designs, you'll find that an optimal design is the best, though in this situation, it's not much better than response surface. The exciting part about optimal design and sequential design is you can use literally half the experimental effort and get almost the same performance. So you can get a lot more out of your experiments if you know what you're optimizing for. I want to add, though, that experiments are not the only solution, though they are the best solution. So since I've struggled to convince people to do these experiments, I've tried to do a crude attempt at estimating these from observational data. So this is a time series from a lake of the cell density of one particular species over about 20 years, estimated once every 2 weeks or so, I think. And you can see there's like 2 to 3 orders of magnitudes of variation in cell density. And along with this, we also have time series of the temperature, of the nutrients, of the light, of the predators. Now, we think that the growth rate or the change between time points is driven by just that very set of factors. So in principle, we ought to be able to back out what these response surfaces are from this data. So I've been playing with this for a while. It has potential. I'm not gonna say that it. I'm really still hoping that people will do the experiments, but till then, this can tell us something. So this is a machine learning inferred 4-dimensional response surface. So just look at one panel with me. Here, this on the top left. On the x-axis, you have temperature. On the y-axis, you have nutrients. And the color variation represents variation in the model predicted growth rate. So what you can see is basically a saturating response with nutrients, as we expect. You see an increase with temperature. It doesn't come back down because, unfortunately, this lake does not just doesn't get that warm. So we mainly capture just the increasing part of the curve. So that's what one light level. As you increase the light, all the growth rates go up, and then they come back down a little bit, which is what we expect. And as you go down, you're increasing the predator density. So as you increase the amount of predators, the growth rate is also going down. So this is all broadly consistent with what we expect and what we know from experiments. I'm not saying that this is truth, because observational data always has challenges, as do machine learning algorithms supplied with limited data. But there is the potential here for us to try and estimate these response surfaces for all the species in a community simultaneously if we have the appropriate time series. Now, it's going to be difficult with species that don't have such simple life histories, but there are possibilities for us to connect observational data with these models that go beyond the species distribution approach. So, to conclude, we need more than temperature projections, because resources and basically every other driver as well shape temperature sensitivity. We don't know yet, but there are probably pretty important consequences for ecology, for evolution, for behavior, and for ecosystem processes. We will not know whether that is true or not until we A, get better data for which there are better experimental approaches, and B, start to explore this in modeling efforts and think about the kinds of feedbacks that are likely to play out. If your primary producers are experiencing this complicated temperature resource interaction, and your consumers are also experiencing a temperature food interaction, then the dynamics of both levels together becomes quite complex, and these are things that we ought to be exploring a lot more. So with that, thank you all.
Question?
Yes.
Would the optimum design venture be the best form of training data for machine learning? Or does that not correspond?
So the optimal design is telling you, if you are doing an experiment to measure a response to some driver, and you have an equation that a function that describes the shape you're after, what levels should you measure? Should you be putting your experimental units at? Until you have that data, I don't know how that could play into the, no, the functions can play into the time series fitting, but the designs themselves do not, because you're basically constrained by whatever the environmental conditions are in the observational data. You don't have control over that. The one thing I have been I've been discussing with people who work on mesocosms, who do these, they have 20 different mesocosms where they turn the temperature knob and a nutrient knob and whatnot, is to basically do something like this, but in that experimental framework. So we have this combination of experimental manipulation and sort of natural fluctuations. Does that answer your question?
It's so far outside my wheelhouse as much as anything to this, so thank you.
So yeah, so if you have any driver in mind, whatever organism or whatever thing you work on, optimal design is just a way of saying, where should you put your effort into measuring things? And it turns out that where you should put your effort is quite different from where we often put our efforts in ecology.
I'm curious, so you made this point that some of these sort of marginal relationships between one variable and fitness have these very characteristic shapes that we can have some confidence in. So I'm curious then, when you're doing this observational learning, why not start from that information as opposed to a machine learning relationship?
So ideally, if we had confidence in the equation, then I would get rid of machine learning entirely. So essentially, machine learning has power from its flexibility, but that comes at the cost of needing a lot more data. So the more assumptions you can introduce into any modeling effort, the less data you need, because your power comes from assumptions. So I started off with this machine learning approach simply because I don't have confidence in being able to write a 4-dimensional equation that can describe that. I think we can only develop that confidence from well-done experiments, but as soon as we have the experiments and we can write those equations, I would jettison the machine learning entirely and switch to state space models.
Yeah, so just a quick follow-up. So I agree that we probably don't know what the interaction of those curves is like. Is there some way to use our knowledge about what they look like in one dimension though to some constraint?
So I didn't talk about it, but I have done that. So at least I've put in directional constraints for nutrients and for predators. So I've said that adding, I have limited experience with machine learning, but there's some algorithms for which you can enforce a simple constraint like that. I haven't been able to figure out how to enforce stronger constraints like unimodality. If I could enforce a unimodal constraint on the temperature relationship, I would. But it is at least heartening that I get a response that's consistent with data even without enforcing that constraint. Do we have time?
Yes.
Yeah. Jen?
Thank you. That was super interesting. I have a question. It was just some of the thoughts you left us with about going up a trophic level, or the dynamics between trophic levels. And I guess when I picture two species competing and driving the resource level down, then you get to the zero net growth isocline, and one species might win the other. So there's no growth because of zero net growth, but there might still be population, like the carrying capacity is not as unrelated or is its own value. So
right.
You're going up trophic level, is it the stock or the flow of your resource?
That what? Can you complete the question?
That is going to matter to the next trophic level up.
Well, at any particular time point, the stock is what's going to matter, but the growth rates are what are changing the stocks. So I think that's where the interaction will come in. Because, so imagine a situation where the predator suddenly consumes a whole lot of the prey. By consuming the prey and excreting, it's changed the nutrient environment, which could shift the environment from one which is beyond the temperature that the species can tolerate to one which it can. So without the temperature changing, it can shift to a situation of negative population growth to positive population growth. And with how that feeds back onto the predator, I think is an open theoretical question.
Yeah, I agree.
and one I wanted to pick your brain about.
Can I ask one more question?
Yes.
So I think when we first started thinking about the fundamental niche, these early drawings were always that they had sort of square boundaries, right? And I think your work is a nice example where we start to see this curved boundary.
Yeah.
And how common do you think that is as we evaluate relationships between multiple different effects? Because those curved boundaries mean that applying stress to both axes, is doubly bad in some way for the population that we're considering. Is that sort of curvature a generality? I mean, I think in niche modeling landscapes, we often think about the convex hull kind of approach, right?
Yeah.
But your work really gives us a glimpse into whether that's a reality or not.
Oh, that's interesting. I suspect it is real for most things that interact with temperature. When you're looking at other pairs of drivers, I am less certain, and I would need to think about. There are certainly physiological reasons why you might expect that with pairs of nutrients, but I would need to think more carefully about specific pairs of drivers. Like there's some things we think about in phytoplankton like salinity, and it's not necessarily obvious why there should be curvature between, say, salinity and a nutrient, though it may be. But I think we will see that with a lot of the temperature interactions.
All right, thank you. All right, well, thank you all for sticking with us. It's a pleasure to introduce Colin Kremer. Colin is here on short notice, but that doesn't mean
you calling me short.
He wasn't initially on our hit list. Colin did a postdoc here at Yale a number of years ago, and so as a member of my lab, I automatically excluded him from the guest list. But he's here today and helping us to round out our program. So it's really fantastic to have you here, Colin. Colin's work obviously interacts to some extent with some of the work Mridul has talked about, but Colin has begun exploring other avenues related to the plastic and evolutionary responses that we see mainly in the microbial realm as well. And so I'm excited to let Colin sort of round out the story by talking a little bit more about the historical legacy effects that we see in individuals and the way that they respond and reshape their thermal performance curves through that lens. So thank you, Colin.
Excellent. Thank you so much, Dave and Martha, for the invitation. I'm really happy to be here with all of you to bring some moderately caffeinated energy to our last session today to convince you how cool phytoplankton are, continuing on from Mridul's talk, and to tell you about some of the things that we've been thinking about that make our understanding of thermal performance curves more complicated, but ensure some level of job security for our next generation. So I want to start with this broad observation that environmental variability is really common, right? So we're seeing variations spatially, temporally, in things like snow cover or the productivity of terrestrial and aquatic environments. We can see shifts in seasons, things that are happening outside the door right now. So we live in a variable world in many different respects. And one of the cool things about biology is that organisms have involved some incredible mechanisms that help them cope with variability in their environment, whether we're thinking about temperature or other factors. I've got a lizard in here since Shane couldn't be here to talk about lizards. So there's a lizard. We even have a mammal here with cool physiological adaptations to help it get rid of excess heat. We've got some plants because plants are awesome, and a tardigrade because who doesn't love those? These all have arguably some really unique, cool biology that helps them do well in the environments, in the variable environments that they inhabit. But one of the unfortunate factors of our time is that our world is changing. So this is an animation of variation in sea surface temperature across different regions of our planet, showing some seasonal and spatial variability in ocean temperature. But we also know, based on historical records and now with the power of our sort of global climate models, that predicted sea surface temperatures are increasing, patterns of variability are changing. And one of the consequences of this change, not just in the marine environment, but also the terrestrial
I guess I didn't drink enough coffee, environment, is that many of these organisms that we care about that already have these cool adaptations may be pushed to their limits, right? As just a few examples, we see that warm adapted tropical species are facing challenges as environments get warmer than any other environment has been on the planet for long periods of time. We see examples of that where species that are facing increased temperature variability may be challenged as well as they start to encounter extremes that they're not prepared for. And this may also affect species that have fewer opportunities to escape from challenging environments if they inhabit locations where there's less heterogeneity in environments where they might find refuge. So in light of this sort of setup, my premise today, starting from this observation that environmental variability is common and influences ecology really at all levels, but starting with the organism, is that we have to understand the capacity of organisms, not just to be affected by their environment, but to react to their environment, what allows them to cope with their the variability that they encounter, with evolution and phenotypic plasticity being sort of high on the list of ways that organisms respond to their environment. And understanding the limits to that or their capacity to adapt is going to be critical to understanding their future in this changing world. So this is especially true that as we as humans continue to alter the patterns of environmental variability worldwide. So this is a real challenge, right? How do we actually go about linking environmental variability to ecology, to phenotypic plasticity, and even evolution, right? This is a really interdisciplinary question as we go about this effort of trying to anticipate the future for many different organisms, critical organisms. So what I'm going to do today is to not solve all of that. I'll save some of that for you to figure out. But in particular, I want to share some examples from my own work, collaborative work with Mridul and others, focusing on phytoplankton, and temperature, thermal performance curves, to try to understand how phenotypic plasticity and evolutionary adaptation affects the structure and shape of thermal performance curves, and hence the ability of organisms to survive in different environments. All right, that's our goal. How we're going to tackle this today, I want to start off by talking a little bit about some background on phytoplankton thermal performance curves, what we know about them, and then we're going to branch out. We're going to think about what we can learn about evolution experimentally and its capacity to change the shape of thermal performance curves. And then we'll also think about some short-term effects, acclimation and phenotypic plasticity, and what that does to thermal performance curves. All right, so what do you need to know about phytoplankton and thermal performance? Well, why phytoplankton in the first place? Riddell covered some of this already, but some of the additional reasons I get excited about it. We might care because they're fascinating organisms, but they're also really important organisms. They matter both in terms of individual species. So Trichodesmium here in your upper left is a species responsible for the vast majority of natural nitrogen fixation that happens out in the oceans, which are often nitrogen limited. So we care about the ecology and evolution of that species in particular. We might care about species that have negative effects on ecosystems as well. So species that might cause harmful algal blooms. We're interested in their biology particularly. But we also care a lot about the bulk properties of entire phytoplankton communities. So as the foundation of the majority of aquatic food webs, that productivity, their growth is really critical to higher levels of the ecosystems that they are part of. So if we're interested in things like food security and fisheries, ultimately we need to know things about phytoplankton and how they're affected by their environment. And then again, sort of reiterating points that Mridul made, we are interested in their productivity as well because of their intimate involvement in global biogeochemical cycles, including carbon. We know that these amazing organisms, these amazing important organisms are encountering many dramatic changes in their environment. This includes temperature, but also changing patterns of acidification, sea ice coverage, and this kind of suite of changing multiple stressors. And that's something that I'm really interested in, but I'm not mentioning today because Riddell tackled that part of things. So I'm going to focus really just today on temperature and phytoplankton. So if we think about what a thermal performance curve gets us with phytoplankton, we can measure their population growth rate as a function of temperature, doing experiments in the lab. We might get a shape that looks a lot like this one here. And then we can start to recognize the key features of that. Where is the optimum temperature? What is the maximum growth rate that this species might achieve? And that lets us do really cool things to make predictions. So we might be able to look at the range of temperatures where this population is capable of growing at all. Thumbs up. when it gets too cold or too warm, this population might not be able to survive. And we can start to actually take these quantitative measurements of these features and connect them up with mathematical models that help us make predictions about the world. So a classic one here would be exponential growth, where your growth rate R depends upon not only your current environmental temperature, but your optimum temperature, maybe which controls the shape of your thermal performance curve. And we can use that mathematical model to make predictions. So under some temperatures, we might see that population growth rate is negative, and we would expect that population to die out exponentially. Under happier environmental temperatures, we might see exponential growth in this microbial population. That's pretty cool, but we can do more. We can start to think about making predictions in time as well, acknowledging the fact that temperature fluctuates in many locations. So we might be able to take some known or measured seasonal pattern of variation or even daily cycles in temperature variation, link that up to our measured thermal performance curve, and make calculations about growth rate over time. So as the environment is fluctuating up and down, the population is moving back and forth across its thermal performance curve. We can aggregate that information to say something perhaps about what the average growth rate of this population was in the environment the variable environment that it encountered, okay? That's cool. How far can we take that? We can go further. Yes, we can. So we might have a measure of this for a particular species, like this marine diatom in the lower left. And then we can actually absorb information on patterns of temperature variation, not just in a single location or a single year, but across the surface of the world's oceans, right? And for each of those locations, we can start to make calculations of when and where the average growth rate of this particular species is positive. That would lead us to sort of mechanistically predict that this species might occur in that location, right? We've heard other examples of that through the talks today. So what I've shown on this map here, the yellow dot there is where this particular diatom was first collected before it got measured in the lab. And in pink here, I'm shading in the regions of the ocean where we would predict the average growth rate of this organism to be positive, right, based on its thermal performance curve. That's really awesome. Then we might ask ourselves, how well does this reflect reality? Always a good question to ask. The gray dots that I've added to the figure now are locations where we have information about the composition of phytoplankton communities, which species were found where. And what I'm going to add now is the location where this particular diatom species has actually been observed. Right? So those are the red dots. Now, this is the group participation part. Did we do a good job making predictions? We've been challenged to make more predictions as ecologists. Here we are, we've got some predictions, and now we get to find out who's an optimist and who's a pessimist, right? So did we do a good job?
Yes and no.
Yeah, yes and no, okay. You're right up the middle, okay. So what's one thing that the optimists like about this? The North Atlantic, yeah, we're doing pretty darn well up here. That's great, there's lots of data right there. That's actually near to where this particular species and genotype was collected. Yeah, okay, yeah. Who cares about the rest of it? I don't know, I want to go out on the limb and make that claim, but certainly if those were the locations where we were interested in making predictions, we'd say, okay, this is doing pretty well. How about a pessimist? Over in here, yeah, there's a bunch of places where it's been observed where we didn't predict it should be. Yep, that's a problem. The Southern Ocean, yeah, it's, you know, there's a couple of instances where it occurs, but it's possible it's called a different species down there, honestly, right? There's some of these challenges with working with global data sets and taxonomic data sets. So we asked ourselves, are these good predictions? I kind of feel kind of in between as well. This is the challenge of confronting beautiful theory with actual data, but it also challenges us to do better. So the next part of our group activity now is to think about what's missing. What did I leave out? And there are a bunch of hypotheses that have been mentioned over the course of today. So this is the pop quiz part of your Climate Day experience. So what did we leave out?
Local adaptation.
Local adaptation, yeah. No evolution here.
Ocean currents.
Ocean currents, yep. I did nothing to talk about the currents that move populations from one place to another.
Light?
Light, yep. Nothing about light.
Nutrients.
Nothing about nutrients. Just left out like Mridul's whole talk, which is not to say it's not important. Yeah. What else? Yeah, there's no higher trophic levels here.
Phylogenetic.
Whoa, yeah. None of that. Nope. Cool. That's a pretty good list. I think that's most of the things that I had on my list. So we've left out anything other than temperature. There's no species interactions. We haven't paid attention to genetic diversity that might lead to differences in phenotype across different genotypes of this species of diatom. I haven't really put any detail into their physiology besides just that thermal performance curve. And I haven't talked about evolution and plasticity, which if you were paying attention to the title slide is what we're going to spend a lot more time on this afternoon in particular, okay? So that's where we're going. If we want to try to understand the distribution of these organisms, their ecology, their response to variability, we need to be thinking about some of these other factors, and we'll talk about evolution and plasticity today. All right, so onwards to evolution. Some of the major questions in this domain we might ask ourselves are, do thermal performance curves evolve? How quickly? What changes about an organism as it's adapting to different thermal environments? And are there any constraints on its ability to make that adaptive response, these organisms, right? How do we go about answering those questions? We might do comparisons of phylogenetic niche conservatism, maybe. As a microbial biologist, one of the cool things that we get to do is actually experiment with evolution in the lab. So that's what I'm going to talk about a little bit now. We can actually keep these populations, expose them to different environmental conditions, and observe how they change in the lab over dozens to hundreds of generations. So the general setup for one of these experiments is we might start with some ancestral population where we know where it came from, what its historical environment was. And then we might expose that population or subsamples of that population to some new environmental conditions. So maybe we increase temperature from 25 to 31 degrees Celsius, and then we propagate it at that generation, at that new environmental condition for many generations. And then we can observe it over time. And we can start to draw comparisons between those evolved lineages and the ancestral lineages to understand what's changed about these organisms, right? So this is a really powerful tool that lets us ask questions about the genetic and phenotypic changes of organisms in response to their selective environment. We can start to also understand things like how quickly did that adaptive response occur. So do phytoplankton thermal performance curves evolve? Perhaps not surprisingly, the answer is yes. These are microorganisms with short generation times, huge population sizes. If you hit them with a strong enough selective pressure, you can get their populations to evolve. You can see changes in their thermal performance curve. And so there are now a collection of examples where people have done these kinds of experiments in the literature and found that you can both decrease the preferred temperatures or increase the preferred temperatures of various species over the course of hundreds of generations, even over multiple years. So that's really encouraging, right? That means that evolution is a potential mechanism of coping to a changed environment for these microbial populations. Of course, a key thing that's been pointed out several times in the literature is the actual rate at which this adaptation occurs relative to the rate at which the environment is changing is going to be really critical for understanding which populations are actually successful and using evolution to track a moving environment. The extra thing, the sort of new twist that I want to put on this to tell you about today is to think about under what conditions we might actually expect this to occur in this world where multiple facets of the environment are changing at the same time, right? So the experiments that I just told you about were all focused on temperature. What happens if you increase or decrease temperature? Do these populations adapt? But we know, particularly in aquatic environments, that an increase in temperature is often accompanied by a decrease in nutrient availability because of the stratification of the water column. So we might ask ourselves, does the outcome of adaptation actually depend not just on the selective pressure of a change in temperature, but also on whether or not that adaptation is occurring with a plentiful supply of nutrients or under a limited nutrient environment? So that's the experiment that my colleague Maria did with this subtropical diatom. She took an ancestral population that had grown for a long time at lab conditions at 25 degrees Celsius, and she performed an evolution experiment imposing a 31 degrees Celsius treatment and looking at these populations to see whether or not they evolved. And under plentiful nutrients, she indeed saw that response, consistent with some of those other data sets graphs that I was showing you. So in black and gray here, we see the ancestral and the control strains, their original thermal performance curves. And in green, I'm showing you new thermal performance curves from these populations after 200 generations of selection. So you're seeing that they can grow now faster under the selected environment at 31 degrees Celsius. That's in the red box. We've also seen that they've got the ability to grow at a previously lethal temperature over here at 34 degrees Celsius, right? The green lines are positive. But one of the cool things about this experiment is we actually have time series information on the abundance of these populations at this previously lethal temperature at 2 different time points over the course of this evolution experiment. What we're seeing halfway through the experiment at 100 generations is if you take populations from their selected environment at 31 degrees Celsius, you start growing them at this previously lethal temperature, the population of most of the replicates dies out in the first several days. And then we see these U-shaped recoveries. These are actually so exciting from a theoretical perspective because it's consistent with predictions from evolutionary rescue theory, that you might have a population that's not able to survive this environment, but some rare genotype that's present in low abundances that can grow in this environment. And we're seeing essentially evolution rescue this population based on that standing genetic variation. So we see this occurring halfway through the experiment. Towards the end of the experiment, you take these populations, you expose them to this previously lethal temperature, and they start growing right off the bat. One way of interpreting that is basically these rare genotypes have now essentially swept to fixation in these populations. They can grow in this challenging high temperature environment right away. You might be wondering to yourself, where are the nitrogen limited curves on this graph? And if you miss them, that's because they're not there. One of the reasons that they're not there is that they actually did not evolve this capacity to tolerate this previously lethal temperature, right? They didn't make it. So if we look back at conducting that same manipulation where we're imposing a heat stress on the populations that were evolved under nutrient-limited conditions, we see at the end of the experiment, they can't make it at 34 degrees Celsius. They're not showing the growth that we saw for the other evolved lines. Even though if we look back in time halfway through the experiment, there's still some evidence that that rare genotype was hanging out in the population. But by the end of the experiment, it is no longer present in high enough abundance or even at all in order to let this population be rescued under that heat stress, right? We've been able to support this with some mathematical modeling as well that seems to tie this back to the idea that protection from extreme heat may actually be due to a nitrogen costly mechanism. So when you have plentiful nitrogen, that strain that is maybe protected, able to grow at higher temperatures, can use that mechanism, increase in abundance, and potentially sweep to fixation. In contrast, under nitrogen limited conditions, we might see a different, basically, thermal performance curve characteristic of that species under, or that strain under limited nitrogen that prevents it from sweeping to fixation by the end of our experiment. Okay, so what do we learn from that? Do thermal performance curves evolve? Yes, in some cases quite rapidly for these microbial populations. It's going to depend a lot on how quickly their environments are actually changing. And this evolutionary response may depend upon what else is changing about the environment that these organisms live in, right? So nutrient limitation may impede their ability to adapt in response to environmental change. Okay, onwards to phenotypic plasticity now. As a very brief review of what phenotypic plasticity is, it's when one genotype produces different phenotypes in response to different environments. We often see that characterized as a reaction norm. This has been studied extensively from the perspective of evolutionary biology. A nice example would be maybe a change in coat color for snowshoe hares under different seasons. And evolutionary biologists ask questions like, when is plasticity adaptive? What constrains it? What enables plasticity to evolve? Whereas ecologists are generally more interested in questions like, does phenotypic plasticity enable invasion or colonization of new environments or communities? How does it affect species interactions or responses to environmental change? But there are also a set of assumptions that ecologists commonly make about phenotypic plasticity. Often we assume that it's beneficial, that it helps organisms, that it's adaptive. This is inherent in most of the examples that we might provide ourselves for why we would see extra defenses or different shaped leaves or coat color. We love our sort of our adaptive stories. Ecologists also assume that phenotypic plasticity is often rapid. This acclimation process is fairly rapid. This was actually embedded in the example that we looked at previously, right, where we're looking at using that thermal performance curve to predict species distributions. Because the thing that I didn't tell you about how these are typically measured in the lab is that each population is acclimated to their particular temperature condition for multiple weeks before this growth rate measurement is made. So we're giving the organisms time to adjust their physiology, whatever they need to do, to sort of acclimate to their particular environment. So this is an acclimated thermal performance curve, and then we use that to predict these species distributions as we're moving sort of across these different thermal environments. So I sort of wanted to ask what happens during that multi-week period where the populations are acclimating. This is work that I actually started while I was a postdoc with Dave, because it's inherently a set of short-term experiments, which is ideal if you're a postdoc or graduate student, right? And so we asked ourselves for this charismatic green algae, Chlamydomonas, can we address some of these common assumptions? Is acclimation actually rapid? Is it beneficial? And what are its ecological consequences in variable thermal environments? So is it rapid? How do we figure that out? Here's a measurement of the acclimated thermal performance curve for this organism, following these sort of standard experimental procedures. If acclimation is instantaneous, as we take populations from different backgrounds acclimated to cold or warm conditions, and we start to move them around, expose them to variable conditions, they should, under this assumption, achieve identical amounts of growth, right? Because they can move instantaneously along that curve. We did this experiment. We took those populations, moved them back and forth between different temperatures, and observed their growth rate. And what we found, under the classic prediction where we can move instantaneously along that curve, we'd have a single predicted final density based on exponential growth. But in reality, our populations with these two different acclimation histories performed very differently, right? This is on a log scale. We can't just predict population growth in a variable temperature environment knowing that single acclimated thermal performance curve. I think this is what you referred to as a disappointing nuisance or something like that this morning, right? Or job security. Is acclimation actually beneficial? Do you gain any advantages as a population acclimated to a cold or warm condition? Our approach here was to try to take these cultures that were acclimated to these different conditions and expose them to different temperatures acutely over short time scales and measure what their growth rates are to see how the shape of those acute thermal performance curves might differ from the acclimated ones. So we did that in these sort of reciprocal experiments. We could measure the growth rate right away. And we get these blue and red curves respectively for the cold and warm acclimated populations. And you might notice that they differ quite substantially from that acclimated thermal performance curve. And in fact, sometimes lead to higher growth rates. That surprised us so much that we did the experiment like 4 or 5 times and spent a lot of time arguing. But this is a consistent result. So we're seeing that these acute and acclimated growth rates differ from each other, and that this has both beneficial effects. Over on the left-hand side there, we see that a warm acclimated population moved to cold temperatures increases its growth rate up to that black line as it's acclimating. So it's benefiting from whatever it's adjusting about its physiology. But we also see the opposite occurring here. The cold acclimated population moved to a warm environment actually grows way faster initially than it does a matter of a week later, right? So it's slowing down as it's coming back down to that black line. So we are seeing that acclimation can be both beneficial and non-beneficial to your population growth rate, depending upon your prior history of exposure. And these are fairly persistent differences. So is it always beneficial? Nope. And this is going to be a thing that we have to pay attention to if we're trying to predict the population dynamics of these organisms in variable temperature environments. So it's great to show that there's a problem with something. It's way better if you can show ideas for how you'd solve that problem. So I want to share a few of our ideas that help us understand the ecological consequences of this phenomenon better. So to do this, we need a framework. There's a nice set of ideas in this paper by Russ Landy from 2014, thinking about how fitness is related to different combinations of environment and phenotype. It's a really cool paper. I recommend it. One of the things that's useful for us in this context is that it links together the environment, phenotype, and fitness. As cool as it is, it does assume rapid acclimation. So we wanted to think about how we might advance on this framework. And so the gist of the idea here is to think about taking these acute curves, the acclimated curve, and then starting to think about not just single acute curves, but an entire response surface. So what happens instead if we start to describe these as performance landscapes, where now we've got acute temperature along the x-axis, the history of exposure along the y-axis. Any given experiment is a slice across this space, our hot or cold acclimated treatments there. The fully acclimated is a slice along the 1-to-1 line. And on top of this, now we can start to plot heat maps or surfaces to understand how different combinations of past and contemporary temperatures affect the growth rate of these organisms, okay? This is a thing that we can do with microorganisms, although it's still a ton of work. But what can we do once we've quantified the shape of these surfaces? We can see where there are high points or low points, but we can also start to combine these with information about patterns of temperature variation. So if we take a time series for how temperature might fluctuate in a particular aquatic environment, we can write down a set of differential equations that capture the idea that temperature perturbations are going to move you in the acute direction. And that physiological plasticity, the acclimation response, is going to tend to move you back towards that 1-to-1 line. So we can look at the interplay between environmental variability and physiology and start to actually look at the trajectory that populations might take through this space as they're encountering a variable environment. And we can contrast that where acclimation is gradual, where you're exploring more of the surface, with what would happen if we assume that organisms could only move along the 1-to-1 line, which would be instantaneous acclimation. They're changing in response to every perturbation immediately, all right? So we have these sort of two contrasting ideas about how the biology of these organisms work. They're just basically moving us around in different landscapes, kind of like that Landy paper. So we're trying to figure out the ecological consequences. We might set out then to perform some experiments where we impose different patterns of variability on experimental populations and see how well can we predict growth rate of these populations. So we did that using different patterns of temperature fluctuation where we controlled the amplitude and the starting temperatures that our populations experienced. And then basically I'm going to contrast our predictions of growth versus our observed growth for this model, assuming that instantaneous rapid acclimation versus gradual acclimation. So here are the results. Again, we've got our observed values here relative to our predicted values. Anytime those line up on the 1-to-1 line, you're really happy as a statistician or modeler. Anytime there's a deviation, right, that means that there's a discrepancy between our models and our observations. And so the take home here basically is that when we use this model that accounts for acclimation as a non-instantaneous process, where we've measured the fitness consequences of different patterns of past and current environmental conditions, we can do a much better job of predicting population growth rate under these different environmental regimes, okay? We have some ideas as to a mechanism of this. This is based on work by one of my postdocs. And the basic idea here is that different factors are limiting growth at low versus high temperatures. So the basic theory goes like this. If you're acclimated to cold populations, your growth rate is limited by temperature, but you have the opportunity to stock up on snacks, basically, right? These organisms can accumulate excess resources from their environment and save them. When you move to a warm environment, when there's an environmental perturbation, that constraint on growth posed by cold temperature is alleviated, and you've got a ton of snacks with you, so you can grow really fast. But as you sort of come back into balance and equilibrate, you consume some of that excess nutrient supply, and your growth rate calms down again. This seems to be well supported by an actual attempt to model this mathematically that fits our observed data really well. I think the last point that I want to make about this is that we've measured these response surfaces now for a bunch of different species of phytoplankton, including both freshwater and marine species from different lineages of phytoplankton. And the structure of these surfaces seems to have a lot of generality to it, where we're seeing this sort of response of cold populations moving to warm environments growing very quickly. So this may reflect something general about the biology of these organisms and their ability to respond to changes in their environment. Okay, so what do we conclude from this exploration of phenotypic plasticity or acclimation? It's not always as rapid as we might assume, but there is some hope. There are ways that we can think about how different combinations of past and contemporary conditions influence the growth of these organisms. Those effects aren't always beneficial. Sometimes they are, sometimes they aren't. It depends upon the exact patterning of the environment that they're encountering. And then this phenomenon can have significant effects for the ecology of these organisms and our ability to predict their growth in variable environments, so especially when environmental changes are large or fast or both. And in subsequent work, we've shown that this can affect actually the behavior of phytoplankton, their ability to, in some cases, move in response to different temperature gradients, as well as the behavior of species interactions in communities of phytoplankton. So this, again, is the places that we've been. To offer you a few final thoughts, my argument today is essentially that understanding the capacity of organisms to cope with changes in their environment through both trait evolution and phenotypic plasticity is going to be really critical to getting their ecology right under these times of environmental change. I've shown you a few different ways that we can start taking apart that problem in the lab experimentally. I motivated this by the fact that we want to understand what this means for real populations in the field, although I'll admit we have not closed the loop yet, right? We're still figuring out these effects in the lab, and then the goal in front of us is to start thinking about how this might improve our ability to say something about what's happening in nature. And solving this problem is especially critical as we humans are altering patterns of warming and variability in our environment. All right, thank you for your attention today. Fight it out.
Awesome talk, really enjoyed it. And I really like the idea of thinking about sort of timescales of plasticity. But one sort of thought that's been, in the back of my mind for a while, and I still haven't quite figured out whether I believe it or not. And I'd be curious to hear what you think. Is it possible that there's acclimation to thermal variation itself?
Yes.
Yeah. And how, right? How then do we deal with that?
Yeah, my hope always is that by resorting to better understanding the mechanisms underlying these responses, we can ultimately save ourselves a lot of work. So if we are able to verify this mechanism that sort of rests on nutrient storage versus the effects of temperature on different sort of patterns of cellular growth, then we might be able to step back from needing these sort of parameterized surfaces where they're measured under particular assumptions about temperature regimes and actually make predictions about the behavior of these populations under novel patterns of environmental variation. But ultimately, if you keep having dependencies, not just on one step back, what was your temperature, but 3 steps back, 5 steps back, the size of the experiments grows inordinately. I think probably however clever we get about our experimental design.
This is what we were hoping to get at with some of the glycerol statistics, right? And so looking for selection on some of, once you kind of know what pathway you're looking at, then you can look for selection on it.
Yeah. And the fact that we see sort of repeatable structures to the shape of these gives me hope in that regard. Maybe it's false hope. Maybe there'll be, experiments slaying the beautiful theory, but we need to do them and find out.
Yeah, totally.
Yeah.
Thanks.
I think you're in line.
I have 2 questions, but I'll just ask one for now. Okay, so you were saying that, I guess I want to push back against your interpretation that sometimes tell me if I'm saying this right, the plasticity is not advantageous. And that was the example of when you went from cold, acclimated, and the warmer temperature, they had higher growth rate at first, and then it came down. And I thought you were going to say, oh, well, that's because high growth rate has a cost. And then go into some literature about how high growth rate, there's a cost at maintaining a high growth rate. So it's not evident. So then I was like, oh no, wait, but in plankton experiments, we sort of assume that growth rate is fitness. But so am I interpreting your interpretation?
So part of what we're latching onto here is actually sort of a long-standing debate in the plasticity literature over acclimation and whether it's beneficial or detrimental. And there are people who have found, it's detrimental. And there are other people who found, it's beneficial, and then they fight, right? And one of the implications of the structure of these surfaces is that depending upon the treatments you use, you may find beneficial responses and you may find detrimental responses.
But by you're saying beneficial, you mean faster growth rate.
Yeah, sort of defined narrowly as does it improve their performance or reduce their performance as they're acclimating. Yeah. From a more holistic perspective of the biology of the organism, we might care about multiple measures of the organism's performance besides population growth rate. So it's sort of a terminology defined in the context of this sort of batch of literature. And one of our arguments is, depending on the treatments you use, you're likely to find a mixture of responses. And so the fact that people are arguing about this may mean that some of them did this kind of experiment and some of them did that kind of experiment. And without taking a look at the larger landscape, they're just sort of looking at different pieces of the elephant, if you know what I mean. Did work elephants in.
Is there an asymmetry in the rate of acclimation from low to high versus high to low? I would think there would be, because we're getting cold, everything is slowed down, so that should slow metabolic rate, slow responses. Except, and also there's the risk factor. If you're warm, and then warm, and suddenly you're cold, what's the chance it's going to get warm again? These are constant temperature experiments, but in the real world, you do get fluctuations. So
yeah, so the surfaces are measured under constant temperatures to try to reduce this dimensionality issue. We don't have any direct experimental evidence that acclimation rate is itself temperature dependent, but I would be shocked if it isn't. One of the challenges is trying to be able to measure growth rate on a short enough time scale to capture that effect. But we've got some ideas in that regard.
Fish data on that,
yeah.
And it's clear.
I mean, to the extent that acclimation is changing what proteins you're expressing or, you know, part of your physical makeup as a single-celled organism, it's got to be temperature dependent. Yeah. Let me know if there's...
Great talk. I was really curious about this idea. I think it came out in 2016. Cameron Gallenbore's work on the role of maladaptive plasticity. And I was curious if you had any evidence of that, if you're familiar with it, and if you had any evidence of that experiments.
Yeah. So in the work that I presented today, we've been very careful to sort of partition evolution and plasticity, in part because we want to know, like, what is the plastic response? So we worked with single genotype populations to do these experiments so that we could, as much as possible, rule out evolution as an explanation for what we're seeing. But the reality for these organisms and many others is that evolution and plasticity both happen, and they're both relevant, and understanding how they interact is really important as well. And if I could yak at you for another 20 minutes, that's the last third of the talk, is to look at what happens when we think about the evolution of plasticity. And if I'm just like on my stump, part of what matters is the pattern of variation that organisms are going to adapt to. Because really, to be fit, to be adaptive, it's what's the pattern of variation they're experiencing that's going to determine what the optimal shape of this response surface is. And when we're just cherry picking pairs of environmental conditions, that's maybe why we're inclined to see beneficial or detrimental responses and then overinterpret them. Because really, they're organisms adapting to a complex environment.
I guess to be more specific, in the population that went through genetic rescue,
yeah,
that was this particular genotype, right? That was one genotype.
It was a mixed population. It was low genetic diversity, but not a single genotype. And in fact, I think this tells us that there must have been a rare genotype in there that could drive the recovery.
But I guess kind of if you looked at like at the initial populations, if you were to measure plasticity, I'd be curious to see if you saw more maladaptive plasticity in this population with the genetic rescue, resulting in rapid evolution, more rapid evolution.
Yeah. That would be phenomenal to look at. This was an exercise in sort of data archaeology for me. So the experiments were done sort of before they brought on a more statistician modeler type person, which is me. I said, oh, could we go back and do this, do that? The experiments are done. But I think that's a really cool question to start thinking about before we even set out to do some of these evolution experiments. We're getting past the question of can organisms evolve or not. We know that they're going to evolve, but to ask some questions about how that interacts with plasticity or other environmental factors, I think that's sort of the next generation of experiments to be doing. Yeah, cool questions. Been biasing this side of the room, but I ignore them.
Kind of a basic question about like the mobility, the dispersal, and the lifespan of these organisms, because I've been thinking here how beneficial is at the end the plasticity for them in the wilderness. I know these organisms go up and down in the water column, so the temperature has actually changed a little bit. But in the wilderness, like having this plastic response that at the end can lead to evolution, is it really beneficial for them? Is it really going to change in these fluctuation levels?
Yeah. So these organisms, I argue with people a lot over whether or not phytoplankton exhibit behavior, but I maintain that they do. So you may not be able to see, but on this adorable Chlamydomonas, there are two delightful little flagella there. And so they can actually swim about their environment. And actually, one of my collaborators did a cool experiment where she created a temperature gradient in a horizontal parcel of water and observed the distribution of Chlamydomonas along that temperature gradient. And she could show that things like the past exposure to temperature influenced how quickly and where they moved along that temperature gradient. So there are definitely opportunities for these organisms to select some aspects of their environment, especially probably in smaller bodies of water where there are stronger temperature gradients, both in space and time. What that's going to do to populations overall is going to depend on not just what an individual cell is encountering, but what their populations are encountering in that environment. Yeah, cool questions. Carly.
I think this is maybe a basic question, but I'm a little bit confused about what is acclimation or like plasticity in some response, and what is the difference between something like an actual physiological response and a more transient response related to like internal as sort of just a lagged effect?
Yeah.
Or is that just a pedantic different?
I think they matter. I think they're difficult to pick out from the literature because terminology gets confused and confusing. So acclimation is generally thought of as a reversible sort of short-term phenotypic response. I've heard people parse that into making a distinction between sort of active versus passive responses. So your measure of performance or traits might change in a new environment even without you changing anything about your makeup, just because under that environment you, say, perform differently. So I think there's a cool paper showing that you can measure reaction norms for beer cans under different temperature environments, right? They're not responding, they're not changing their physiology in response to being in a new environment, but you can measure things about them that are different and hence construct a reaction norm. So that was kind of a cautionary tale to think about. What about changes in your organism in a new environment are due to actual active changes on its part to regulate its physiology to that environment versus just things that are different when you look at it in a different environment. And I think in some respects, this nutrient storage may not, I don't know the extent to which it's an inescapable reality of their biology versus something that they're actively regulating. But I think it's a good question that you're asking. Yeah.
Oh, no, I'm just curious. So you mentioned that these phytoplankton can move and exhibit behavior. So I was just curious if you, or what other metrics of performance are used in phytoplankton besides growth rate, like to make thermal?
Yeah, great question. It's becoming slightly more common for people to try to decompose that a little bit more into what's happening at the physiological level. So we can start to do things like look at changes in respiration versus photosynthetic rates at different temperatures. In my lab, we're starting to do more work with flow cytometry and stains to actually try to tease apart birth rates versus death rates in these populations. That's pretty exciting as well. So sort of continuing the theme of taking ideally simple organisms and recognizing more of their complexity. Yeah.
Are there any energetic costs to extreme?
Probably. I mean, it takes energy to use a flagella to move around.
Capacity to maybe do that?
Not to any substantial degree. There's still very small organisms that can get trapped by things like turbulence or boundary layers. So they have some capacity to move. Others do it by more like regulating their buoyancy, perhaps, but they are still largely at the whims of their environment.
So they wouldn't try to do it. I'm trying to see to the extent that, for lack of a better term, almost able to reason through it, like they wouldn't be fighting. Is there some sort of optimization or opportunity to this in that they would try to move when conditions suited, or they'd be completely unaware and they'd be trying to fight that and failing? Or?
Yeah, so some of the strongest gradients in aquatic environments are vertical gradients. So the light is at the top, The nutrients are at the bottom. You need both of those to make a living as a phytoplankton. So there's some interesting work sort of trying to figure out where should a population sit in the water column to maximize its growth. You could certainly throw temperature in there. And then to any extent that they had the ability to regulate their depth, they would presumably do that to try to maximize their growth rate. Their ability to do that is going to be strongly limited by the realities of existing as a single-celled organism. But
with respect to movement, a number of years ago, John Heron did an experiment, and unfortunately never published it. But he put volvox in a gradient with a light down here, and the volvox would move towards the light.
Yeah.
Photosynthesis. They do that at a whole variety of temperatures, but once you get the temperature to a certain level, they move away from the light.
Ah, cool. That's phenomenal. Volvox is like the best name ever. If I ever have a bowling team, it's going to be Volvox Fierce Rollers.
And with that,
Thank you.
so I'll just remind everyone that we have got a poster session that'll kick off in about 10 minutes upstairs. There's going to be food and snacks and drinks upstairs as well. There are photos that were submitted by a number of members of the community that are on display. So I invite everyone to stick around, socialize, ask questions of our speakers, and enjoy the rest of the afternoon. Is there anything more we should add, Martha?
I think that's it. I just wanted to thank the speakers again and all the participants for coming today.
Yes, thank you, everyone.